according to the American Cancer Society, the lung cancer is the most common cancer in both the lady and the gentlemen in the USA. I believe that's also true in the most country around the world. And the lung cancer can basically divide into two subgroups. One is the lung small cell carcinoma, the other is the small cell carcinoma. My topic is mainly focused on the first group, the lung small cell carcinoma. For the etiology, the smoking still is a major cause for the lung cancer. There's also other factors that's no related to smoking. And so what's the thromboxane? Thromboxane is the molecule catalyzed by the thromboxane synthase. And then there's also related to the arachidonic acid and the COX. So the function of the thromboxane has to the interaction with the receptor. There's two types of receptor in human. First, there's a TP alpha, the other is TP betas. But in the mouse, only one receptor is TP. So why are we going to study the thromboxane, the pathway in the lung cancer? Because the function of the thromboxane, there are several functions. First, it can promote the proliferation, and also promote the angiogenesis, and promote survival. All those features is quite close, similar to the tumor, to the tumors. The previous study also demonstrated level of the thromboxane B2. There's the stable metabolized thromboxane A2 is significantly higher in the non-small cell carcinomas. So this study, there are three questions we're going to ask. First, what are the level of thromboxane synthase and downstream products in the lung cancer? So we're going to measure the thromboxane in the lung tissue sample from the cancer, or also from the lung cancer, from the lung tumor lung tissues. The level of thromboxane B2 is much higher in the tumor when you compare to the lung tumors. And there's also demonstrated the level of thromboxane is significantly correlated with the stage of the lung cancer. That's just in the higher the level of thromboxane B2, the more advanced stage of the lung cancers. So the second question is any relationship between the smoking carcinogen and the thromboxanes. First, we have to know that smoking that can generate dozens of the carcinogens. Among those carcinogens, the NNK is the most common one, also most powerful one. And the NNK has demonstrated can induce the lung tumor in all the animal tests so far. So we use the NNK to induce the tumor in the mice, also use NNK to treat the cells. And this the model we use to establish the lung tumor, use the NNK. After NNK injection, about around 34 weeks, we can see the lung adenocarcinoma develop in the mice. And then around 38 or 40 weeks, 100% of the mice have the tumor in the lungs. And we also monitor of the level of thromboxane synthase in the lung tissue. We found that the level of thromboxane synthase increased around 34 weeks. There's also and then we measured the thromboxane in the tissue sample from the lung patient with the lung cancer of the smoke. Also from the patient without the smoke. We found that level is higher in the smoke compared to the lung smoke. And that's the immunochemical, the staining of the human lung tissue from tumor and lung tumor, also from the smoke and the lung smoke. The level of the synthase is higher in the tumor compared to lung tumor, also higher in the smoke compared to lung smoke. And in the cell line study, we treat the cell with the two types of the lung epithelial cell with the NNK. We found NNK can induce the expression of thromboxane in the dose-dependent manner, also in the time-dependent manners. And NNK can stimulate the cell to produce thromboxane. And when we add the thromboxane inhibitor, that's a fibrillate, we can find the NNK fail to stimulate production thromboxane B2. So this result in the thromboxane synthase is responsible for the NNK induce the production thromboxane. And the third question we're going to ask, how does thromboxane contribute to the lung carcinogenesis? So we're going to measure several cell functions. First function we measure is the cell proliferation. We use MTTSA to measure this proliferation. When the cell treated with the NNK, we can see the cell proliferation is increased. That's in the time-dependent manner. And then when we inhibit the thromboxane synthase, we can find that the cell proliferation is decreased. When we another chemical is called SQ29548, that's the thromboxane receptor antagonist. We can find that also can block the NNK effect on cell proliferation in both cell line testing. And that's just another method to measure cell proliferation is the BRDU in covalent nuclear to measure. We got a similar result. And then we also measure another marker, pCNA, that's the marker for cell proliferation. The NNK can stimulate the marker. Once the thromboxane was inhibited or its receptor was blocked, the effect NNK disappeared. In this experiment, we use another chemical called U46619. That's the chemical can directly stimulate the thromboxane receptor. So we can see this marker can synchronically promote cell proliferation. It's as powerful as NNK. This effect cannot be blocked by thromboxane synthase inhibitor the inhibitor that's fibrillate, because their chemical is a direct stimulate the receptor. So when you inhibit the synthase, there's no effect that's blocked. This is you measure the apoptosis with the tunnel assay. We found the NNK can inhibit apoptosis once the thromboxane was blocked by its inhibitor, or its receptor was blocked by its antagonist. The apoptosis increased. By measuring molecular, we found that the NNK can increase the BCL2, decrease the BAX. Also, the marker of apoptosis, cleavage PARP, was decreased by NNK, but increased by the thromboxane synthase inhibitors. So this result indicates the inhibition of the thromboxane or its receptor can induce apoptosis, probably by decreasing BCL2, increasing the BAX, and cleavage of PARPs. As mentioned before, in the human cell, there are two types, the thromboxane receptor, TP alpha and TP beta. So we're going to check which the receptor is contribute to the NNK-induced lung carcinogenesis. First, we check the expression of those two receptors in the cell line. We can see the TP alpha can expression in the expression in the all type the cell line testing, but the TP beta, some of the cells did not express these receptors. And then we measure the expression of these two proteins, the receptor protein, we found TP alpha was increased in the tight pair manner, but TP beta did not increase by the NNK. So this resulted in NNK induced TP alpha protein in the tight pair manner, but no TP betas. So we also measured the mRNA of those two receptors as well as the thromboxane synthase. We found NNK can stimulate the expression of TP alpha mRNA, but no the TP beta, but also cannot stimulate thromboxane synthase mRNA expression. We also measured the two kinases, AKT and the ERK, also one of those tumor suppressed gene PTEMs. We found that NNK can induce the ERK and AKT, but inhibit the PTEMs. Because the PTEN is controlled by the microRNA 21, we also measured microRNA 21. We found NNK may inhibit the PTEN by the enhanced microRNA 21s. This the experiment to further study the pathway related to the NNK induced lung carcinogenesis via the thromboxane pathway. We found that we use one chemical SN50, which is an NF-kappa inhibitor. That this result demonstrates NNK activation is necessary for the NNK to induce the thromboxane, but thromboxane synthase, but not the receptor TP alphas. And then we also use the kinase inhibitor. One is the U0126, which the ERK inhibitor. The other is the watermarine, is the AKT inhibitor. And the result demonstrated the ERK, but the node AKT function upstream the NF-kappa to participate NNK-induced thromboxane synthase. And then we also studied the COX-2, use the inhibitor of the NS398, which is a COX inhibitor. We also, and the other inhibitor is the SQ29548, which is the TP antagonist. We found that the TP activation can stimulate the COX-2. Also, NNK-mediated thromboxane production depends on the presence of the COX-2s. And that's the experiment to use the TP, there's a receptor, thromboxane receptor agonist, to see the TP, the role in the NNK, that mediate the cell proliferation. So we use the chemical U46619, that's TP agonist. So we found that those results indicated the inhibitor COX-2 by its inhibitor NS398 or sRNA can increase the ERK. And the bottom part result is that indicate the inhibitor COX-2 can prevent the separation, and this action can be blocked by the TP activation. The TP activation is done by adding the U4619. So there's the experiment. We used those chemicals. One is the actinomycin, which is well-known, the transcriptional inhibitor. The other, cyclohexamide, is the general translation inhibitors. So the result demonstrates NNK-induced TP alpha while translation rather than the transcriptional mechanisms. We also checked the thromboxane synthase, the 3' UTR. We found there's the binding site for the microRNA, the 34B and 34C. So we further demonstrate NNK can inhibit this type of the microRNA, and we further demonstrated the microRNA, the 34B and C can function as thromboxane inhibitors, and NNK can increase the thromboxane by decreasing the microRNA 34 B and Cs. And last question we're going to ask, what's the best strategy to target the thromboxane pathway? So there's the on top here, top panel is the traditional pathway. There's the well-known, demonstrated by many different studies. And the bottom pathway is a newly identified pathway in this study. That means that once the receptor TP-alpha is activated, can further go into active COX-2 and then down to activate the thromboxane synthase, and finally the thromboxanes. That's a newly identified pathway. That's a very important step is the root is the TP-alpha autoactivation pathway. We have done a lot of experiments to get the sub-conclusion. I'm not going to go through the very detailed those experiments because time is a limit. So the first sub-conclusion is NNK, the initial the unalkable signaling depends the TP-alpha, but no TP-betas. So TP-alpha can enhance the unalkable pathway. And the second sub-conclusion is that TP-alpha can increase unalkable mediated thromboxane and COX-2, but decrease the P10s. And the final sub-conclusion is inhibit the TP-alpha can attenuate unalkable mediated cell growth, and therefore TP-alpha stimulate cell growth. So in there's the brown color. There's the autoactivation pathway identified in these studies. In this pathway, we have a lot of steps can stop this pathway. For example, we can use some small molecule such as SQ29548 or BN567 to stop the activation of the TP-alpha. We can also inhibit the COX-2. There's a lot of COX-2 inhibitors available in the market. And we also can inhibit the unalkable, and finally we can also inhibit the thromboxane synthase. We can regulate the thromboxane synthase by the microRNA 34 B and Cs. As previously mentioned, the pathway, the thromboxane is controlled also by the COX-2. So when we hit the COX-2, we also can inhibit the thromboxane. But once we hit the COX-2, there's probably can produce a lot of side effects because COX-2 control a lot of sub-pathway, not just thromboxane pathway. So it's better to target the thromboxane synthase. That will probably reduce the side effect. So the conclusion is that first is the level of thromboxane and it's molecular. It is metabolized. Thromboxane are increased in lung cancer. And the second, TP-alpha or the TP-beta promote the tumor growth, and NK increase TP-alpha. And the third, NK induced the thromboxane synthase can be inhibited by the microRNA 34. And for TP-alpha play a positive role in the NNK-induced COX-2 unalkable activation, leading to the upper regulation of thromboxane expression and the thromboxane productions. And the fifth, the newly synthesized thromboxane can further activation TP-alpha, forming a auto-regulation feedback loop for TP-alpha activation. Finally, TP-alpha and thromboxane synthase are ideal target against the smoking-related lung cancer. Thank you.
Great. So questions from the floor?
There are many gene mutations in lung cancer. KRAS among the Caucasian or FGFR in the Asian. Any correlation with your thromboxane?
Yeah, there's a lot of gene mutation. The in the lung cancer, in the subset of lung cancer, like EGF alpha. There's also the well-known those inhibitor available for the patient to target those kind of patient. So I think our pathway identified here is different from those the mutation pathway. Though I haven't checked very detailed how many percentage those patients have those kind of mutation. But in my assumption, this pathway is different from previous identified pathway that's available, those EGF alpha that's available and inhibitor available in the market for treated patient.
So
once a target or an antagonist for the thromboxane synthase is identified, speculate on how this might be delivered to the patient. Would this be a systemic administration or perhaps a pulmonary administration to get a localized effect at the lung?
We haven't had the data in this area, but I think locally apply those inhibitors locally to the lung probably better than can reduce the side effect. But we haven't figured out how those going to be in the future then.
all right, well, thank you very much. I think we're ready for lunch. We're going to break for lunch and see you here after lunch, which will be, I guess, at 2 o'clock. Enjoy your lunch.
Thank you.
And if you don't mind, so this is, you want to take over here.
Okay.
There is this young research forum.
There's also us?
Yeah, yeah.
There's also us?
Can you just do everything? Because I have a poster session I'm supposed to go and,
Or you want me to take everything from you?
yeah, is that okay?
Yes, okay.
Yeah.